Welcome everybody to another round of Sensitive Scientific webinars. The topic of this webinar is the physiologically based biopharmaceutical model report template. It is my great pleasure to host this webinar. The usual host, Oliver Hatley, he couldn't make it. I can see him, he joined, but he had to leave early, so he gave the responsibility to me. And it is my great pleasure to welcome Summit Aurora from Johnson & Johnson. He has kindly agreed to take us through this report template. Summit, if you go to the second slide, please. Yes, many of you, you know Summit. He has been in the field for several years. He's now senior principal scientist and at the J&J. He received his PhD in biopharmaceutics. And for many years, as I said, he had been working in the area of biopharmaceutics, oral peptide, and now focusing more on the PBBM. Over the last several years, a concerted effort have been worked on the area of PBPK, PBBM, and regulatory side of those. I'm not going to explain anything more. I leave it to Summit to take you through all of the history and where and how did we come to this template, which is a very important and essential part of work that Summit led the work to get to this point. It was a collaborative effort, and as I said, it's going to take us through. For your information, we are going to record this one, and it's going to be available on YouTube shortly. Summit has kindly gave us the permission to record it and make it available on YouTube. Feel free to ask your question, please. If you have got any question, either raise your hand or put your question in the chat. And I think we can leave either the question to the end. It depends on how Summit is comfortable. Or we can give a pause at certain points, and then we can go through the question if there was anything, or we can leave all to the end. I don't know. How do you want to do it?
For me, that's fine, Masood. We can make it interactive, so please feel free to ask your questions in between as we are going through. So
okay.
Seems good.
Okay,
yeah.
so yeah, I will keep an eye on the hands that they are written or the chat. Okay. So yes, I leave it to you, Sumit.
Okay.
The floor is yours.
All right. No, first of all, thank you, Masood and team at SEMSIP for providing this opportunity to talk a little bit about the work which we have been doing in the field of PBBM and the report template which Masood has mentioned. Idea for today for me is to give you a brief overview. How did we get started? What did we do to get here? And this work has already been published, so you can find it. It's in a virtual issue. And I'm going to share some of the links later as well, which you can refer to. So yeah, I like to start just with the acknowledgements first. It was a huge effort, and I'm just a voice on behalf of this big team who led this work together, along with multiple industry partners. We have 4 regulatory bodies along with us who were helping in shaping the template. Of course, the software companies, Satara, Simulation Plus, were also part of this ones, helping us making sure that all the insights from their sites are captured in the template as well, and together with our academia partners, M-Cersi at University of Maryland. So yeah, this is just a quick overview around what is PBBM all about. It's physiologically based biopharmaceutics models. It's a subset of PBPK, which is used for oral formulation development. So here you can really see the areas which are highlighted in black are the applications for PBBM. So that's really around the quality of your drug product. It can be used to support virtual bioequivalence between different formulations as they are being developed or refined during the drug development process, trying to understand what is critical bioavailability attributes of those formulations. So really understanding your critical material attributes, critical process parameters, which then has an impact on dissolution and therefore the PK performance. PBBMs are also being used to set quality specifications. For instance, API particle size specification is one example. Dissolution safe space is another example. And it has also been used to use to do bio waivers if, for instance, there is a change in the site, or if the F2 failure is happening, then there might be an over discriminatory dissolution method, which then can be supplemented with PBBM to try to justify why you don't need to run a BE study. If, for instance, on long-term storage, we started seeing some of the batches going out of stability. So a lot of applications of PBBM are there for CMC, but there are other applications which are related with PBPK in general, which is DDI, special populations, pH mediated DDIs, food effect. But the focus of this template has been really on the CMC sections, more on the drug product quality. So there is very different experience among the different health authorities, and it became clear when we interacted with the health authorities that some are much more advanced compared to the others, and some are much more used to see the submissions compared to the others. There is no common standard by which industry should use a template to submit it to the agency. They see kind of different informations coming through to them from different channels. Only FDA has issued a PBBM specific guidance. There is a guidance on PBPK from EMA, but FDA is the only authority which has guidance specifically for PBBM. And when we got together in 21, this group of industry, there was a huge variety of experience across the industry, and there was not a consensus like why certain models were rejected, why certain were accepted, and so there was a need to bring everything together and try to do this a little bit more systematically. Okay, how do we go from there, and how do we make sure that the regulators are also up to speed with what the industry is following? So several benefits of PBBM, we can use it to improve the mechanistic understanding of the drug product. We try to understand what are the key drivers which are impacting the PK. It can be either process related or materials related or formulation variables. It can help us understand the edge of failure for your CMAs and CPPs, and therefore you can get more flexibility in setting your drug product specifications while approaching the PBBM rather than going the conventional route where you just put the specs as per the batches which you put in your phase 3 clinical trial. Post-approval changes is an important aspect for PBBM. And yeah, if there is a changes in manufacturing side or deviation in the formulations, we can use PBBM to justify bio waivers and gain that regulatory flexibility. So quite some applications of PBBM. So just give you a quick background, where do we started with? This is almost in September 21 when this group got together. Where the idea is that, okay, we have been to a lot of PBBM conferences in the past, and there also what was happening is like the industry presents the case studies, there were discussion happenings in different breakthrough sessions, regulators share their overall feedback on the submissions they receive, but we never received very concrete feedback in terms of what regulators like, what they didn't like, what they would like to see. So we thought, okay, let's turn the table around, and in the workshop which we organized in August 23, the idea was that the industry prepares a set of case studies, and you can see the name of different companies here and the case studies which they prepared, and submit these case studies to the regulators. And it was also a great commitment from the regulatory side, because that's coming on top of their day job, that they committed that, okay, if you submit these case studies, we will review it, and then during the conference, we will give you the feedback, why we like this study, why this was accepted, why this was not accepted. So it was a really nice conference, and a lot of good discussion happened in that conference, where we gave the stage to the regulators and actually listened to them. Okay, what are the pain points they see in these case studies? Um, and there was also a very strong commitment from the software companies as well, because they agreed to train the regulators for assessing the submitted models. A lot of regulators, they don't have that background or that educations to evaluate the models, but then the software companies committed as well to train them. It was also agreed that they can take help from academics, so they can onboard some academicians to help them evaluate. And then and what also happened during that evaluation is, like, regulators at the background were also arranging meetings among themselves to then try to learn from each other experiences and also discuss their findings. So at the workshop, all of this information was presented. We actually had 16 foreign regulatory agencies which was participating there, 22 academics, a lot of industrial and in-person participants were there. So if you want to have a look at that, there is a summary paper around what happened at that workshop. There were 3 days. There are individual manuscripts covering each of the 3 days. And now there is also the PBBM template, which came out from that workshop. So quite a productive couple of years, really moving the needle in the field of PBBM regulatory acceptance. So there was a survey, and we looked at, okay, what are the top 5 reasons why we started seeing a lot of rejections of the PBBM? And these options came, these points came out from that. So it was an inadequate model parameterizations. There was inadequate model validation. There was lack of agreement on the proposed validation criteria. So regulators were not really sure why a certain criteria was set up. Models did not include non-BE batches. That regulators would like to see some kind of failure mode, that the model can pick up what is acceptable and what is not acceptable. But we know from the industry side as well that it's always not so easy to make a non-BE batch. So there were discussions around, okay, if you are in that scenario, what should we do? And then, of course, some models which were submitted where the dissolution method was not biopredictive. So there was not a link between in vitro dissolution to in vivo PK predictions, because the in vitro dissolution is a key link there, because that's what you measure on your drug product. And then you try to link it with the model to try to predict PK. So these were quite some criteria, and you can already see that, okay, parameterization, validations, lack of agreement, this is something you can address it if you properly have a structured way of providing that information to the regulators. So we did ask a question at the workshop, and you can really see that everybody agreed that there is a value to develop a PBBM report template, because you can imagine if everybody across the industry started submitting these reports in the set format, and regulators are getting used to see this information flowing in a set format, it will enhance the review process, and could have a positive impact on the field. So we said that, okay, this was a mandate there, and there we then the group got together, and over a year and a half work, we got the template published. So you can find the template. It's available as a Word document, which can easily be downloaded directly from the supplementary information. But we also have a manuscript which is published along with the template, and the manuscript discusses key themes which came out when the template was getting reviewed by different industry and regulatory members. So we did also submit it in the supplementary information in Excel file, where you can really see over 400, 500 comments. Sorry, go ahead.
I don't think it was a question. It was more of a noise. Sorry about that.
Okay, no worries. So you can really see also in the supplementary information in Excel file, where we, for the sake of transparency, we wanted to share that how does the review process has happened, where you can really see the comments coming from industry and regulatories and how those was addressed. Because when you get 400, 500 comments, it also takes time to how to make sure that each of these points are adequately addressed and a common resolution is made in the template. So what we did when those comments were coming in, we tried to structure those comments in common themes, where these comments were coming in, and these themes are discussed in the manuscript, which could be the good topics for future conferences and breakout session. So we made it clear right at the start that this is not a harmonization exercise. This is not a regulatory guidance exercise. This is more really coming from the scientific perspective that the template aims to summarize a set of expectations and it proposes a structure uh, for structured data input, modeling plan, parameters, and clinical data, and how you are showing your model development, verification, and application. So we really approach it from the scientific point of view, considering what is the best practices there and how these models should be submitted. It is very descriptive. It's the template itself is a 40-page document. It has very different sections, and and there are guidance provided under each of the sections, which the sponsors and which the sponsors can use to guide them what information might go in those sections. And of course, it's a not inclusive template. We have a lot of varied applications for PBBM, but we try to make it as general as possible. But of course, depending upon your question of interest and for what the model is being developed, you can customize the template. But it did serve as a point, like if you see all those points in the template, it does give a reminder that, okay, if certain parameters is not relevant to your model, you kind of explain it and don't assume that regulators already knew about this. So that's really the intention why what we said right at the upfront, for what purpose we want to develop this template. So it consists of 8 different sections. You have a regulatory executive summary. I'll go a little bit detail on bullet point 2 and 3 because I think that's an interesting one. How do you really frame around your question of interest, context of use? How do you assess model risk and regulatory impact? These 2 points, 2 and 3, also forms the basis of draft guidelines, which has come from ICH M15. They also talk about this. So what we also tried to do, because we have some regulators who were part of the ICH M15, and they were also part of this effort as well. They really helped us, keeping us in a track and in line with what they were going, what ICH M15 is talking about. So if you think about ICH M15 guideline out there, this is more around like a what. So it's a major framework. And PBBM is a very specific example which can fit it into that ICH M15 guideline. So the ICH M15 explains this frameworks of around question of interest. And if you really want more tangible example how to actually apply ICH M15, I think the template is a good resource there. And then, of course, there are rest of the sections which talks about materials and methods. That's the biggest section in the template, talking a lot about the model parameters and how it can be parameterized. Of course, the results discussion and conclusion then. So just for your quick overview, this is how the different sections in the template it looks like, and a lot more detail you can find it in each of the sub bullets mentioned here. So just going forward, I won't go into a lot of details of the template, but I would like to focus a little bit more on assessment of the review process. So I already talked a little bit about how we organize that in different themes, and going forward, I would like to share those themes with you and the common points which came during the review of the template. So one of the common point which came was around that different people were using different terminologies. So for instance, if you look at the US FDA guidelines, we have the terms like verification, validation, whereas EMA talks about qualification. So there was no agreement on these terminologies, and we also don't wanted to have that agreement within the template, but fortunately we have the M15 coming along. So we tried to what we tried at the time of the template was to harmonize the terminologies which M15 were proposing and try to adapt it for PBBM specific application. And that worked actually quite well. The comments we got from some of the regulators which were involved for ICH M15 draft guidelines is that really fits, this template really fits under ICH M15. So that kind of, what we needed here is like we agree on the terminology and we move on, and we say, okay, this is what does this mean? And regulators understand this, we understand it, and let's just get the point across and agree on it and move on. So we did provided a glossary in the publications, which sponsors can copy paste in their reports, and we try to make this very much aligned with what's there outside with the ICH M15, but also FDA PBBM guidelines and EMA PPPK guideline as well. So it's a good resource to be included in the reports. I'm going to spend a little bit more time on question of interest, context of use, model risk, and regulatory impact. I think those are the concepts we knew exist, but we are not discussing this structurally in the reports. And ICH M15 provided that framework, and we thought, okay, yeah, this makes sense that this is the first things regulator would like to see when you start writing these reports. So you clearly specify on your first page, what is the question of interest for which a model is being developed? And then the context of use talks about what is the specific role and scope of this model is in order to address the question of interest. I'll show you an example later to make this concept a little bit more tangible, but it really talks about is your model is the major piece of evidence in the entirety of data, or it is a supportive evidence compared to if you have clinical data or some preclinical data. So how do you position this model? And this gives ability to the regulators to really then say, okay, is this a major piece of evidence? Then we need to make sure that this has been evaluated with more stringent criteria. So that's where the regulators would like to see what is the scope and role of this model to address the question of interest. And this can also be used to then assess what is the model risk. And this is just one way to frame model risk. There is currently no alignment. How do you go about describing a model risk? There are multiple ways to do this, but we thought that Kumil et al. Has published a nice strategy around assessing model risk. So this is just one way of doing this. So you can divide the model risk into two buckets. One is model influence. So again, what is the role of the model in the totality of data? If the role of the model is minor and it is only explaining certain observations, then of course the model influence is low. But if you are trying to use the model to weigh a clinical study, then of course the model influence becomes high. Then there is the term called decision consequence, which looks at what is the consequence of that. If your model is wrong, are you putting patients at risk? And certainly in the PBBM framework, yeah, we are talking about bioequivalence limit of 80 to 125%. So CMC questions are usually of less risky for the patients because we do have most of the time quite wide PK/PD understandings, not for all drugs, but for majority of them. So if your PE trial is failing at Cmax, yeah, it doesn't pose a risk to the patients if the efficacy is driven by, let's say, AUC as an example. So it does bring that context also to your model risk, like, okay, if the model is failing with these and these margins, what is the decision consequence? And then based on this model influence and decision risk, you can make 3x3 metrics and then try to explain your model risk and classify it in that way. So that's one thing. And again, this is the model impact where you say, okay, it evaluates the role of the modeling in the decision making, and you can clarify that, okay, this is low, medium, and high. And together with model risk and regulatory impact, would determine how strict the verification and validation criteria should be. So this is a topic definitely for ongoing discussions. How do we assess this objectively? How we come to the model risk, and how should then the verification and validation criteria be set up? So as an example, this was one of the case studies which was submitted by Johnson and Johnson, and here what we were struggling at that time is we had a regulatory approved quality control dissolution test. And what we started to see that when the drug products was stored under long-term ICH stability, some of the batches were going out of specification from the dissolution test. So of course, one of the questions which we wanted to see, okay, can these out of specification batches be approved based on virtual bioequivalence to the reference product? And how can we use this model to then justify the widening of quality control dissolution specification? So the context of use, so if you have two questions, you also need to clearly specify two contexts of use. So the first one is that, yeah, we used model to perform VBE to demonstrate bioequivalence between an out-of-spec batch and a reference batch. Therefore, we want to eliminate running a clinical study. That clearly shows that model is a main piece of evidence here. And then for the second one, we submitted the package, the entire package along with the model to try to justify that our current dissolution is over discriminatory and maybe we need um we need to extend the boundaries of the dissolution safe space to get more flexibility. We think the model influence is medium, but for some, I already got some comment in some of the earlier webinars that this is, of course, you're trying to mitigate a clinical study, it's high, but we say it's medium because we have quite some clinical data to already build confidence in the model. We already have a non-bioequivalence patch to make sure that the model is able to capture between what is acceptable and what is not acceptable. Decision consequence was low because we had a wide PKPD. And we know that the patient safety is not impacted if we are failing at a certain confidence intervals. The geo means are passing, but if the constant confidence intervals are failing. So it was a low, medium model risk, and the regulatory impact we also thought it's medium because we had additional clinical data to show non-BE batches. But that, of course, would be an interesting exercise. Going forward, that if we propose this, how does regulatory see this, and do they agree with our model influence and impact? And if they push back, then how do we go about this? So it would be an interesting exercise to do in future workshops. Another theme was around, okay, how do we go about defining drug product variants, and what should a fit for purpose clinical data should look like? And there the agreement was clear that, okay, the drug product variant should be described based on the variable for which you are trying to set specification. So if you're talking about particle size specifications, it would help if some of the clinical data is generated with different particle size to be then used to validate the model. We always thought, discussed is that, okay, sometimes we try to deliberately change the drug product to try to get a different dissolution profile, but the drug product still needs to be representative of your commercial formulation. So for instance, one of the way to reduce the dissolution rate is to completely take out the disintegrant, but that might change the drug release mechanisms and may not be the preferred way to go about it. So that's something has to be done case by case basis depending upon your drug substance and drug product properties. There is always good that if you have that non-BE batch to do the model validations, and quite a number of models were rejected because they don't have a non-BE batch. But we did talk about that as well, that, okay, if you don't have a non-BE batch and it's super difficult to manufacture one, if you have already generated data at extremes of your process parameters and material attributes, and if you have some clinical data at those extremes, that could also serve as a confidence in the model. And you can use it directly actually to specify justifications without even using PBBM. But what we also thought is, okay, it's not always easy to have those patches, but what is the model is trying to do? The model essentially is trying to link your dissolution to your PK performance. So dissolution is really key in PBBM. And for instance, if you have a weak base which shows a different performance in a PPI condition compared to which is a proton pump inhibitor conditions compared to a normal fasted conditions, you can actually use that data set also to build confidence in your dissolution model. And if you see some kind of PPI effect, that could be used to justify a non-BE criteria, because you are then using physiology as a surrogate to create discriminatory condition. Same goes for a fed state simulations if a drug product shows bile-mediated solubilization. So it's really about generating that confidence in your dissolution model. And lastly, we always don't start with a drug product which is commercially ready. It goes multiple changes as we are moving through the drug development process, from API batches to manufacturing processes for the drug product, morphology changes of the API as we go through the through the through the process. So if some of those clinical data and relative B studies are done with different variants, yeah, try to leverage that piece of information and make sure that you generate that confidence in the dissolution model for the PBBM. All right. Um, Method development and parameterizations again was a major topic there, and there again was the request that yeah, we should clearly outline the source of certain parameters and decision takens around certain values which are assigned to certain parameters. So that should be really clearly defined. Parameter identifiability is a common issue within PBB and PBPK models because there are multiple parameters which you can optimize to get to the same answer. So really trying to do a lot of sensitivity analysis to try to find out how each parameter is impacting the outcomes, and therefore justifying why a certain value is choosing for a certain parameter is a key there. And the template really goes in much more detail here to try to guide the sponsors how they can go about specifying different parameters. Um, Model validation and acceptance criteria is again an important one, and it's an ongoing topic of discussion. Um, we do believe that higher the model risk or the regulatory impact, more strict the criteria should be. Um, and it should be in line with what you put criteria on your clinical studies. So there are several criteria which are proposed, but again, there is no common consensus around which one is the best or which one you should go about. But there were few guidelines around, okay, the prediction error should be less than 20% average across the entire clinical studies. If you are simulating relative BA studies with 16 patient data or 16 healthy volunteer data, 80 to 125 is usually a good criteria. But we also thought that, okay, sometimes we use these models to try to predict what we have observed in our single ascending dose and multiple ascending dose studies. And there we are working with 6 to 8 subjects with 2 placebo arms. So that's a really, really small sample size. And then making sure that you can always be 0.9 of your observed PK parameter, either Cmax or AUC. Yeah, that's not always easy to achieve. And that also determines on the variability which you observe for your drug substance. So a study specific criteria for those kinds of clinical data, if you plan to use it in the model validation, yeah, we can justify that. And then let's put a rationale around why you think for this kind of studies this makes sense. And if you are, of course, using a relative BA or a bigger studies with more number of subjects, yeah, the criteria needs to be a little bit more stringent. Model application, again, we discussed a lot about VBE in that. That's the major application for PBBM. But the question which were discussed there was around, okay, what criteria should we use to determine the number of subjects? And how do you go about parameterizing within subject and between subject variability? So quite some nice work being ongoing and published in literatures from software companies to try to come up with novel approaches around this. There are two ways you can approach it. One is bottom up, where you can propagate your within subject and between subject variability in physiological parameters. That does require two characterization of these parameters, and that's a tricky one. So you really need well-controlled studies to try to figure out this within subject and between subject variability. Another way is to apply variability directly on your simulated PK parameters, but then you are assuming that the variability between a reference and a test is the same because yeah, you just apply a cumulative number on your PK parameters. Lastly, model reuse was another aspect. It was quite an interesting one from some of the companies because, of course, yeah, once we developed and invest heavily, these models requires a lot of resources, and to write those reports, yeah, if you can find a way to reuse those models, that of course is beneficial for the for the company as well. So it really depends that if you are trying to reuse a model and if you're changing your question of interest and context of use, I think it was a general agreement that model risk and regulatory impact needs to be reevaluated. And if your validation verification criteria still hold true, yeah, then you can go ahead. But if you are now approaching a more impactful application of model, then that needs to be stringent as well. So that's the piece where you can redefine it, but rest of the sections like going into and describing all your parameters, you can refer to the previous submissions. Um, US FDA has also introduced this concept around model master file, which really talks about file sharing, acceptance, and communication of the models. And actually, model reuse is also a very cornerstone for model master files. So that's basically it from my side. So I really would like to thank all the collaborators who has worked on this. It was really fantastic. Couple of years, very productive. Um, the paper is out there for all of us to use, and we do hope that it kinds of bring a little bit more um, systematic reporting of these models to the regulators, and therefore try to promote the acceptance of these models. So we'll see in a couple of years once we gather more data and once different companies share their experience of the template, then if we need to revise this in next editions, yeah, that's something we can definitely look into once we start getting some feedback from different stakeholders. Um, so yeah, I talked about these are the 5 publications which is out there. There is actually a virtual issue where you can find all the 5 publications. So it talks about the workshop, it talks about the summary and all the 3-day papers, and then there is a PBBM template there as well. So yeah, please go ahead and have a look at it. It's quite a good summary of the discussions which has happened at the workshop. And yeah, let us know if you have any comments and any question. Um, so that's
Yeah,
actually all from my side. Yeah.
thank you very much, Sumit. Yeah, we have a good amount of time for question and answer. One topic that I didn't want to get in, but we are into it, which is PBBM and the PBBM guidance. If you remember at the PKUK when you were presenting, I mean said that the FDA guidance is not the PBBM guidance. It is a PBB guidance for biopharmaceutical applications. What is your view on that? There were lots of chats on that one, so you don't need to read them.
Indeed. Uh, indeed. I think it's again is yeah, it's a personal opinion. And I agree. Yeah, I agree with people. For me, the crux which we from the industry are working is to get these model accepted for CMC applications. So either it is called PBBM or PBPK for biopharmaceutics applications. I don't think I have a very strong opinion there. Um, what we want at the end is to gain flexibility in how we bring the best product with best processes to the patient. So I think patients is the North Star, and getting those things in time to the patient is what should guide, because the resources we save here is invested for other researchers. So yeah, not strong opinions there, but I don't know, Amen, Masood, if you want to take a crack at that one and share your opinion there.
Yeah, no, I think Divian, sorry, because that one is related to the discussion. So Amin has raised his hand. So let Amin go first, and then we go
Yeah.
back to your question, Divian. Yeah. Amin, please go ahead. You have to unmute yourself.
I unmuted, but it's not working.
Now
Oh,
we can hear you.
Yeah.
Okay. Sumit, I have got a sort of personal views on that. My comment was, if FDA wanted, they could have called it PBBM guidance. And there must be a reason that they did not actually call it PBBM guidance. So my point is that it is misrepresentation of FDA's position when we call this FDA's PBBM guidance. They could have called it PBBM guidance. Why they didn't call it? So we cannot, on behalf of FDA, misrepresent this guidance as PBBM guidance because they never called it PBBM guidance. There is only mention of once, in one paragraph that it's in the discussion, and they basically said, "Oh, some people called it PBBM," but obviously, if they wanted, they could have called it PBBM guidance. So we shouldn't actually call this PBBM guidance. We should call it as it is, and then say, okay, this guidance can be used for all the PBBM work that we are doing. So the point is that, sort of they haven't called it PBBM, and we shouldn't misrepresent, this guidance as PBBM guidance.
Yeah, the essentially the methodology is the same, right? I mean, PPPK, I don't know. Yeah, it's something, frankly, I don't.
Look, if they wanted, they could have called it PBBM.
Yeah, definitely.
You can't basically on their behalf call their guidance PBBM guidance. This is not right.
Yeah.
Okay, Divyan, do you want to go with the question?
Thank you. Thanks, Sumit, for a wonderful presentation and publications. So I really appreciate it. Being interested into PBBM or biopharmaceutics part. I have a few practical questions. So number one question is, with respect to specification. So normally if we see EMA guideline for dissolution specification, they says that for immediate release, normally directly a Q point with a single time point is given as a Q point in a spec, or for a modified release, it is normally 3 time point given for a specification. And now when we talk about the sorry, Amin or Masood, let me tell it PBBM currently. So if we talk about the PBBM guideline and as a user input, what do you suppose whether a spec time point as a dissolution should suffice, or practically if I was the regulator, I will expect entire dissolution profile as an input, and then based on that specification may be supplied. So do you think only 3 time point for modified release or a single or few time points for a specification can be inputted, and then based on that dissolution widening can be approved?
No, it's a good question. Just to give a bit more perspective, how do we handle this from the industrial side of the things? When we submitted and I'm only talking on behalf of innovator drug products because that's what I have experience on. So when we submit these drug products, we do submit whole justification and every complete dissolution profile to the regulators. So they do have access to that information. And then based on your clinical relevance of your QC method, you come up with a spec of Q80 at 30 minutes. I'm just picking a number. So there is a whole lot of clinical batches which goes in your phase 3 trial, and then you use those cloud of batches to justify a spec. That's one way of approaching dissolution spec, that you come up with a Q number, or if you have a modified release, you can come up with 3 points. Another way to do this is with a dissolution safe space, that you don't specify a spec, but you go to the regulators and say, this is my dissolution profile of the mean batches which went into my phase 3, but I have so much information around this mean profile that instead of proposing a spec, I can be 10 or 15% lower than the spec, but I still would be okay. So you kind of go with your dissolution safe space, and then you propose a spec on the lower boundary of your safe space, because that really gives you a lot of flexibility in your life cycle management. So that's the way. We have a couple of examples where you can go either traditional way and do it with a Q spec directly on your phase 3 cloud of data on your QC profile, or you go with PBBM and then define a spec based on your lower limit of the dissolution safe space. So it really depends on the projects, the type of data you have, and how much negotiation you can have with regulators to come up
So
with that kind of flexibility.
yeah, thanks. But it requires a lot of data.
Yes.
But on the same point, why not the upper point or upper, like lower boundary? It should also have a upper boundary because there is also risk of failing on a upper side. And second question is with respect to the failing batch study. So this may sound stupid. So does it require a bracketing approach? It means one batch on a upper side failing and another one on a lower side, or as of now, I know only one batch, whatever side it is failing, it should suffice the need. But does regulators want such a thinking that also a bracketing approach should be there, same way like IVIVC? Because with IVIVC, we can modify the or remove the excipient and every other thing. With PBBM, we are not modifying the excipient or Q1, Q2, basically. So what do you
Yeah.
think? Whether they do require a upper and lower bracketing approach even for a failing, or it is just one?
Yeah, it's usually the lower. It really depends on your PKPD, right? We so if you are failing the lower boundary, what has been impacted? It's efficacy, which you are worried about. That the patients might get lower drug, lower dose, and they don't get the efficacious dose there. So that's what you are saying with a lower boundary. When you talk about upper boundary, you are more worried about safety aspect. Is something related to safety can happen in the patients. And if your drug has wider PKPD, so then you are not so much worried because also in a spec, if you look at traditional how the specs are set, you are always worried about batches following below spec, not upper spec. Upper you always have if a profile is at Q80, yeah, 30 minutes at Q80, if the profile is 85, nobody is worried about that.
No,
So
but
it
that
needs
is
to
the
be,
point that
yeah,
for a normal drug or safe drug, it suffice.
yeah,
But when it is such a drug where safety needed, for
then you
example,
can,
normally ibuprofen is considered to be safe and effective. For that also, like BioWaver article was written by Jennifer, I think so. If we see the same article and based on that, if we consider this will be my last question, I want others to participate, so sorry for that. So if we further see EMA's PSG for ibuprofen, where they are also asking for the statistical analysis for a comparison for a Tmax,
yeah.
do you anticipate that? I don't anticipate that it should have any effect of, but should have an impact with a Tmax, but ibuprofen, which is considered normally safe, effective, there is a. So, what are your thought on that part?
Yeah, yeah, I do have to look into that a little bit more. Um, but yeah, there must be a reason why they're asking for it. So I don't have all the information to make a statement around that at the moment.
No worries. Thank you.
Okay.
Thank you for all the answers.
Siri, do you want to
Yeah,
go ahead?
thank you, Sumit, for presenting this. So my question is on non-B batch. So what do you think FDA's position or EMA's position and other agencies' position on non-B batch? Are they still pressing for having a non-B batch, or did anything change after from the 2023 meeting where we tried to propose to use maybe PPI or FedFX as a way to verify or validate the model rather than just depending on non-B? Did anything change in the agency, or are they still pressing for non-B batch?
I think we have to wait for another conference city to share the experience. I haven't heard anything directly. We don't have the, yeah, we haven't interacted in the meanwhile with them personally from our side, from Johnson and Johnson side. So I can't give you if anything concretely changed. That's a topic of discussion. I think we probably need to send out a survey to the members to get that feedback. But I do think that they were also um, you really have to share the understanding and take the way to approach this would be to share your understandings on your drug product, on your drug substance, and then really make a case why you cannot make a non-BE batch. And for
Yeah.
that, yeah, go ahead, sorry.
Yeah, yes, I'm saying, yeah.
Yeah, so we did had some experience where we showed that critical bioavailability attribute analysis, and we did receive positive feedback there, because then you really demonstrate why it's very difficult to make a non-BE batch, and then you try to substantiate it with others. So it's a very common practice, at least from the industry side, that once you are filing, you really assess critical bioavailability attributes and then try to link it back and say why within your set process boundaries, within your formulation manufacturing space, you don't expect a non-BE batch. So that's how the specification works. So industry do a lot of work in that respect. And just presenting it in the right context to regulators should be able to justify if you cannot make a non-BE batch. If you have it, then I think the problem is solved already.
Okay, thanks for this update.
Okay, Goreng, do you want to go ahead and see? I know you put it as a chat, but you can read it as well.
Hello, Sumit. Yeah, first of all, thank you for presenting it. It was really useful. So, my question is regarding: is there any consensus or a kind of harmonization in assigning the risk levels? Because as you said in your case, that maybe from industry point of view, it's a medium level, but then from the regulatory point of view, it may be a higher risk level or something like that. Is there any efforts going on in that direction? And the second one is more important regarding the acceptance criteria. So how uh, is there any efforts going on from both industry and regulatory side, especially on the virtual bioequivalence acceptance criteria? Because whether we are considering the confidence intervals, and if we are, then is there any criteria on how good it should be compared to the actual clinical observations? And then depending upon the variability observed in clinical studies, like either it's a highly variable drug product or a low variable. So what is the things which are going on from both industry and regulatory point of view? And then what are the future directions in these two areas?
Yeah, no, thanks, Gaurang, for the question. I don't think there is an actual consensus on this, because this is also quite new with the ICH M15 coming in. But what I found it interesting about these concept is that it gives us our side of the story to write it in the reports, and then try to justify why we think a certain accepted criteria should be accepted or should be put forward. And you clearly explain why you think, and bring just not only the modeling aspect, but bring the totality of information which you have for your question of interest, what you have to address your question of interest. So no, to your point, I think Kumal is something which is still widely considered, but there is no set expectations. I do know that companies at the conferences which we met, like GSK, AZ, and us as well, we will be submitting our models going forward with this template in what we have put together. So let's see how does the feedback comes from there, and then we will definitely know more. And regarding your validation criteria, I think this is something, again, case by case specific, very much related to your molecule of interest, to your question of interest. We did had cases where we did submitted different acceptance criteria based on clinical study design, and that was accepted. So I hope that answers your question.
Yeah, thank you.
Yeah, thank you very much, Sumit. Just follow up from what Goreng was asking. That was one of the questions I obviously we haven't had any follow up. How do you see the uptake of the report template? So is it, you mentioned a few companies. Do you see that is, are you hearing other people they are starting to use it or at least adopt it?
Yep, I hear positive comments so far, Masood, but those are some companies which openly said that they're gonna give it a try. And I also know that there is a guidance from NVISA coming up on PBBM, and they're gonna officially endorse template as the way to submit models to NVISA. So that's already something, a positive win for the team who put this work together, because if one agency promotes it, accepts the model, I think this might be the start. So yeah, hopefully we'll see that guideline from NVISA coming up.
Yeah, yes, that's very good. That's very good news to hear. Are there any other activities or initiatives as a follow-up to this effort that
I think we are still looking to see the possibility if we can put this template as an annex to ICH M15, because yeah, ICH M15 is very general. If you guys had a chance to read through it, yeah, you get the concept, but then at least it's on my side and I can share my feeling there. When I read the guideline, I think, okay, it makes sense. It presents the concepts, but how do I go around it? How do I actually apply this on a particular model? And I think there, the template which we have put together is a very nice example to show how you can apply ICH M15 in the field of PBBM. So yeah, that's one of the things we are looking, and let's see if we get that feedback, because there are negotiations going on, if we can go and work on PBBM template as an annex to ICH M15. And then definitely, I think there are still talks ongoing to look for next round of workshops on model risk assessment, acceptance criterias, experience which people may have on the template. So it's still early, but I think in a couple of years something should come along.
Yep, hopefully we will see that. Yeah, and you're right. You mentioned some of the areas, and it was in the question as well. Acceptance criteria, model validations, model risk. These are the elements that even in the current MIDD guide, the draft guidance M15, they are not necessarily very clear, and they were mentioning that they are like what you guys did in the template to come up with some examples to show people how to apply them.
Yeah, we did presented like 6 examples in the manuscript to talk about different areas of PBBM and how you can apply model risk assessment to those. There are some examples from generic as well.
Yes. Any other question? We have 1 more minute left. We can use Summit experience a bit more if there are any other questions. Yes, Goreng.
This is just a general question. So why are we restricting it to PBBM, biopharmaceuticals application, whereas it could have been applied to any model-informed drug development application? So your thoughts on this?
I think it's fair. We restricted it because just to produce, like you can definitely adapt the template, Gorang. I think the structure of the template is flexible, and you can definitely adapt it for another context. Because we were really focused on drug product quality, we really wanted to nail it down how this should work for the CMC space. But definitely, I think if you're talking about dermal model development and other routes of administrations, the template can still be applied to those.
Yeah, just follow up from what Samir said. If you are looking at the structure of the template, it is, I would say, it is very natural way of presenting your data, and it is to a large degree inspired by the EMA guidance and template and what they want, and FDA, because you see that what they want. So the context of use, the question of interest, and then model assumption, sensitivity analysis, model validation, all of these that they are contain. But I would say I give credit to the biopharmaceutic community who made a concerted effort to make it adapted specific application of biopharmaceutics. So, and make it, but as you said, it can be generally applied in different areas. So there's no reason to stop there, and it can be applied in different areas. Yeah. Okay, Sumit, any other comments or final thoughts?
No, nothing from my side. Thank you again for the invitation, Masood, to share this work.
Yes, thank you very much for your time and also agreeing to present. Obviously, it is great to see this work is out, and as Sumit said, hopefully we will see its uptake in very near future in different areas and specifically in biopharm, and we see more acceptances of biopharmaceutical application. Thank you very much, everybody, for joining, and hopefully we'll see this one shortly posted on YouTube. Just watch out for the follow-up webinars on SimSip Scientific webinars. Thank you very much, everybody, for joining.
Thank you.